Good morning. Welcome everyone to our seminar series here in the Center of Biotechnology in the Federal University of Rio Grande do Sul. So today I have the pleasure to introduce and talk a little bit about Gabriel, Professor Gabriel Castrillo from, he's in Nottingham University right now. And he has a wonderful publication record. You're gonna see him talking about some of his more recent work. I'm not gonna give too much details. He's gonna present a little bit about himself before he starts. And I just wanna point out that Gabriel is a very young researcher, a very well-published researcher. He has Nature papers, Science paper, papers published in Plant Cell, Plant Physiology, Current Opinion in Plant Biology. So it is a great pleasure for us to have him here, and I'm very thankful that you could accept our invitation to be with us today. And I'll give you the microphone. You can start. Thank you, Gabriel, for being here with us today.
No, thank you, Felipe. Thank you all of you for this great introduction and your kind words. So for me, it's a big pleasure to be here today, to be honest. So my intention is to share a little bit of the work we are doing in Nottingham. So as Felipe said, and Gabriel, here you have my email in case you want to contact me at the end of the talk or whatever thing you need. And also we have here our website. Essentially, what I want to be doing today is talk a little bit about our main results related with the project we have in our lab. It's basically the description of the coordination between the microbiota and the root endodermis, and how this coordination impacts the mineral nutrient homeostasis in Arabidopsis thaliana. So before I start with the science, I would like to introduce myself a little bit. So I did my PhD in Spain in almost 12 years ago, working on phosphate starvation response. And I learned a lot during this period of time of my life, but not enough to consider myself ready for a PI position. So I decided to do a postdoc working in arsenate signaling. Arsenate is a chemical analog phosphate, so it was kind of in field. And after I finished my first postdoc, I realized I was not fully prepared. So because I was missing the microbiota contribution to phosphate arsenate signaling, I decided to move to United States to the lab of Professor Jeff Tangle in North Carolina at Chapel Hill. And then I spent there another 4 years working in the interaction between plant and microbiota in the context of phosphate starvation. After that, I moved to Nottingham here in UK, and I get a position as a Nottingham Research Fellow, and recently have been promoted to Associate Professor here in Nottingham. My lab is mostly focused, as I said, in plant nutrition, and I want to explore the angle of plant-micro interaction. We have essentially 4 different lines in our lab. One is basically interaction between root diffusion barrier and microbiome. That's the one that I'm going to explain today. The other one is the extraction of principle about the microbiome assembling in the root, using Arabidopsis thaliana as a model. And also an interest in the study in how the micro that is living in close association with the root impact the root development. And on top of that, we have kind of a recent line that I want to explore what happened with the nutrient recycling when the microbiota is colonizing the plant. And with this one, I explore a little bit the crop angle. So it's not only Arabidopsis, but also maize and rice. As I told you, I've been working in the area now for more than 7 years. First, I've been involved in excellent project relating essentially how the microbe and defense coordinate on the phosphate starvation response in Arabidopsis. This work has been published in Nature in 2017. And in this line, I also contribute in another project related with the root development and the microbiota. And essentially, the sum of these 2 projects is the molecular mechanism regulating the coordination, because with this project, I combine root and nutrient, as you can see in 5 minutes. And essentially, what I want to tell you is that sometimes your pathway determines who you are at this particular moment. So, and I want to start talking about science. As you know, the population of microbes that live in close association with the plant is what we call the microbiome. This population of microbes can live in close association with the root, but also with the shoot. And we are learning more and more that this population of microbes is quite relevant for nutrient uptake and use, for example, but also for the activation of the plant immune system. So I don't want to give you a lot of detail. There are massive amounts of paper, but this is something that we cannot ignore. I know that the majority of research has been conducted under axenic condition, but more and more we are aware of this issue, and we are learning and extracting principles of this interaction, and we are learning how to incorporate microbiome to our research. In other words, I think that the microbiome is critical for plant survival in nature. And importantly, this population of microbes is able to colonize the root massively. It is present in every single zone of the root, from the meristematic zone to the mature zone. And also, you can find not only on the surface of the root, but also in the inside, in the endophytic compartment. You can find microbes from the endodermis to from the epidermis to the endodermis. And you think about the localization of the microbe in the root, somehow remind me what happened with the nutrient uptake and use in the root. So in this scheme, you can see the soil here, and this is the vasculature. You see that essentially the root is taking the nutrient and also the water using three main pathways: the apoplastic pathway, the symplastic pathway, but also the transcellular pathway. But in the root, these three pathways coexist until the endodermis. Essentially, because in the endodermis, we have what we call root diffusion barrier. That is a deposition of lignin in the space between the endodermal cells, and commonly known as Casparian strip. But also we can find the position of another polymer in the endodermis that we call suberin, and suberin and the endodermis kind of control the diffusion of water of nutrient, because until the endodermis you can diffuse kind of freely, but at the level of the endodermis, because of this root diffusion barrier, only transcellular transport occur. So in other words, the endodermis is controlling the mineral and water homeostasis in the plant. But at the same time, the endodermis somehow need to exert this function with the microbiota present that is metabolically active. How this process happens is something that we don't know in plants. But in human, I don't know if you heard about it, at the level of the basal epithelium, we have a very nice interaction between the resident microbiota and the nutrients. And there are mechanisms described about how this interaction happens and the consequences. And this is, now we know this as the regulation of the gut-brain axis, with all the consequences for our social behavior, neurodevelopment, and anxiety. So we also know from humans that imbalance in this interaction between the gut diffusion barrier and the microbiota provoke a lot of diseases. But if you pay attention to the design of our gut that is here and the root, we can find a lot of commonalities. Essentially, the absorption of the nutrient in the gut is happening in the upside of our gut, of course, through the hair we have in the gut. But this absorption of water and nutrient is controlled essentially by the time junction and the basal epithelium. But if you go to the root and analyze the anatomical structure of the root, you realize that there are a lot of commonality. We also have hair in the root, and also we have an outside, that is the soil. And at the level of the dermis, we have also a control for this nutrient diffusion and water that we call the Casparian strip and suberin deposition. In other words, what I try to let you know is that plant root is like an inverted gut. So it's a very nice model to understand what happened in the gut and establish similarity. So with this idea in mind, we start thinking that if this mechanism that happened in the gut exists and is already described, probably we can use this information to explore what happened at the level of root. And that was essentially our motivation. So our main hypothesis was maybe in plant we can find similar mechanism operating or controlling what happened between the endodermis and the resident microbiota, and what is the consequences of this interaction for the nutrient homeostasis. And with this idea in mind, we start what we call the cross-kingdom hypothesis. To demonstrate that, how the root diffusion barrier and regulatory network interact, we use essentially two different approaches. One is we need to prove that the root diffusion barrier regulatory network are controlling somehow the assembling of the microbiota. But at the same time, we need to prove that elements of the microbiota per se is able to induce perturbation or somehow control the function of the root diffusion barrier in the root. Obviously, this is not an easy task. So, because the synthesis and the dynamic of the root diffusion barrier is tightly controlled, I don't want to give you a lot of details about how this regulatory network performs, but I want to let you know that it's essentially controlled by a transcription factor called MYC36. And MYC36 orchestrates the expression and the proper localization of several proteins that promote the polymerization of lignin in the case of the Casparian strip, and the proper localization of this lignin between the two endodermal cells, building like a bridge that controls the diffusion of nutrient and water. So this polymerization is mediated by lipase, another enzyme that has been described nicely, kind of recently. And we already have all of this information. And also we know that the integrity of the Casparian strip is controlled by a pathway called Schengen pathway. And taking advantage of this information, what we did at first was to identify a collection of 20 different genotypes. These 20 different genotypes represent different level of impairment in the synthesis and function of the Casparian strip and the suberin deposition. We artificially divide these 19 different genotypes in 7 groups. For example, group 2 have a defective Casparian strip with ectopic lignin deposition and increased suberization. And for example, group 6 have no suberin and it have a really defective Casparian strip. So number 6 group essentially have no root diffusion barrier. So we can cover all the variability from a normal root diffusion barrier down to almost nothing. So what we did was to identify a location in our campus in Nottingham, and we collect soil that had been exposed, I will say minimal to the human activity. And with this soil, we grow our collection of genotypes, and we identify the population of bacteria living in association with the root and also with the shoot. And at the same time, we quantify up to 20 different elements, mineral never in the shoot of this plant. The first thing that we noticed in our experiment when we quantified the rosette area was that the size of the different mutants was different. This is something that we need to take into account, probably because the interaction of the genotype with the soil and the microbiota, or the interaction of all of these edaphic factors that happen in the soil. But when we quantify the actual abundance of the different microbial population across the different genotypes, and this is what you have here, the different genotype and the amplicon sequence variants we identify, we see that there are certain of them that are significantly different across our collection of genotype. And these differences are maintained also at the family level and the class level, suggesting that the microbiome assembling across this collection is quite different from wild-type plants. Here you have a canonical analysis of principal component, and in red I put the one that are more significantly different from Columbia, that is number 1. And you see that essentially the difference across the microbiome is distributed in our collection. So it's not a determined group of genes, it's essentially the whole network, suggesting that the genes that are controlling the synthesis of the Casparian straight and also the suberin are important for the proper assembling of the root microbiome. When we quantify the concentration of the different mineral nutrients in the shoot of this collection of genotype, we also detect difference. So here you have the genotype, and here you have the different mineral nutrients we analyze, and you see that in some cases these differences are quite obvious. Interesting when we compare the dissimilarity found in the microbiome matrix with the ionon matrix, that this is the comparison we have here, you see that there are a nice correlation, suggesting that probably the mechanisms that are controlling the ionon, the mineral accumulation in the shoot, are kind of the same that control the microbiome assembling. In order to prove this a little bit more, to add more robustness to our analysis, instead of working with the whole collection, we identify some of them to cover all the groups, and also instead of working with the natural community, we identify 42 different microbiomes that somehow represent the diversity found in soil. And we move into the agar system. Instead of working with soil, natural soil, now we are working with agar system. And under this condition, if we compare the size of the rosette, we see that across our collection, this difference getting reduced in the agar, so we don't have this developmental effect anymore. Under this condition, we also detect that the assembling of this synthetic community in this genotype is also different. And as you can see here, for all of them, the microbiome assembly is significantly different from wild-type plant. And when we quantify also the distribution of the different minerals in the shoot, we see differences, as you see here in this CAP analysis. And again, we see a very nice correlation between the structure of the matrix for our microbiome and the structure of the matrix for the ionoid, suggesting that essentially not only natural soil, but also on the control condition, the mechanism that control the synthesis of the suberization and also the Casparian strip are the same that are controlling the microbiome assembly. To explore this crosstalk a little bit more and share a little bit light about the mechanism and how these things can happen in the root, we identify a collection of 400 and something, it's close to 500 different isolates, bacteria isolates, from the lab of Jeff, Julia Borhot, and Paul Schaeffer-Lefter in Max Planck. They kindly sent this collection to us. This collection had been isolated from healthy plants, so there are no pathogens involved in this collection. And somehow this collection resembles the complexity of the diversity found in soil. And in order to use the collection and to shed light on how members of the microbiome affect the function of the Casparian strip and the suberization in the endodermis, we designed a couple of screening. Here you have the screening pipeline. Essentially, what we did was, for the case of the Casparian strip, we used a cellular tracer called PI, propidium iodide. So the propidium iodide is a cellular tracer. It can diffuse easily through the apoplastic compartment until they reach the endodermis and getting stopped by the Casparian strip. So by measure the distance between when the Casparian strip stops the diffusion of the PI and the tip of the root, you can have an idea how premature the Casparian Casparian strip is formed, it is. So in the case you have an impaired Casparian strip, the diffusion going to be higher, so you can reach the vasculature easily. In the case of a premature formation of the Casparian strip, this distance getting shorter. And that's the analysis that we did in the case of the Casparian strip. To evaluate the suberization in the endodermis, we quantified the number of cells in the 3 zones that the suberization happens. So suberization in the root happens in a very organized way. You first have a zone of no suberization, and it slowly evolves into a patchy zone. So some cells are suberized and another don't. And he evolved finally into a continuous lamel of suberization. So what we did was, in the presence of an individual member of this collection, we quantified the number of cells in this zone, in the patchy zone, and also in the continuous zone. By quantifying these 2 phenotypes, we can a general idea how the individual member of the root microbiome affect the function of this root diffusion barrier. We did that in the case of the Casparian strip. This is this top panel. In red, you have the values that are significantly different from a no bacteria control, that is this horizontal line. And you see that in the case of Casparian strip, so a propidional diffusion, we identify around 27% of the isolates that have the capacity to modify the deposition of the Casparian strip. Surprisingly, in the case of the suberization, you have here horizontal line with the value of no bacteria control and different color for the 3 zones. So no suberization is in brown, green for the patchy, and blue for the continuous. You see that for some isolates, like in here, the continuous zone practically disappear. So, and for example, this case is quite interesting. There are Ephingomonas. So Ephingomonas have the capacity to remove the suberization from the root. And we was quite surprised about it, because it was the first evidence that the member of the microbiota have the capacity to remove practically the suberization from the root, with all the consequences it have for the mineral, nutrients, and water homeostasis. To get a little bit deep into this mechanism, we select 42 different bacteria, which phenotypes span across all the PI analysis, but also across all the different phenotypes we observe in the case of suberization. And we were able to reproduce a very nice gradient of suberization using these 42 isolates. So every dot in this figure is an isolate. And what we are measuring here is the distance from the root tip to the continuous lamel. And you see that we have nicely reproduced this gradient. So in other words, we have a collection of 42 that somehow contain all possible behavior we found in a bigger collection of 400 and something. And this span from a lot of suberization down to almost nothing of suberization. And the next question was how, by removing or introduce perturbation in this root diffusion barrier, what is the consequences for the accumulation of mineral nutrient in the shoot? So we were able to identify cluster of mineral nutrient that reduce the concentration in the shoot when you also induce perturbation in the suberization. But also, you see here, this cluster increase the concentration of all of this mineral nutrient when you induce this gradient of suberization. In other words, when I try to show you that there are a very nice correlation between the suberin and the mineral nutrient deposition in the endodermis. So for some elements, this correlation is negative, but for some of them is also positive. As you can see here, is the same data. For example, in the case of boron, this correlation is negative. So the concentration of boron in the shoot decrease when you decrease the suberization in the endodermis. By contrast, in the case of calcium, for example, this correlation is positive. We also analyzed that this difference have nothing to do with any kind of fertilization effect. So to prove that, we prepare in parallel another experiment, but this time killing the bacteria. We kill the bacteria and we quantify the ionome again. As you can see here, for the heat kill control, the majority of them grouped together with the no bacteria, suggesting by adding the bacteria per se is not allowed to induce any type of perturbation in the mineral accumulation as compared with the bacteria alive. That is what you have here. So to understand a little bit more this amazing mechanism of crosstalk between suberization and the microbiota present in the root, we designed a synthetic community, essentially because you don't have in nature, well, you have in nature individual isolate, but they are not working individually, they are working together. So we designed a synthetic community of these 42 different isolates together. We proved that these isolates have the capacity to colonize not only the rhizoplane, but also the endophytic compartment. And we exposed our plant to perturbation that we know, nutrient perturbation that we know to provoke oscillation in the suberization in the root to absorb this perturbation. As you can see here, when you imposed minus iron, you see that the suberization is reduced. By the contrary, when you remove potassium for the medium, this suberization is massively increased, as has happened as well when you put a sodium chloride. So yes, this is, at this point, this particular result was published in Cell. I think it was the lab of Nico Gellner. So this plasticity was known, but the new thing here was what happened when you add the complexity of the microbiome to this analysis. Surprised, what we see was like when the synthetic community of this bacteria synthetic community is present, somehow this plasticity disappeared. Now we have less oscillation of the suberization in the root, suggesting that the synthetic community have a buffer effect on this suberization plasticity. As you can see here, here is the quantification of this phenotype. When you don't have the microbiota present under axenic condition, you can see this plasticity. As soon as you incorporate the synthetic community to the system, this plasticity is getting reduced. But what does it mean? The first thing that we noticed was that we have consequences. So when you have the microbiome present, for all conditions, the plants look healthier and the rosette was bigger. As you can see here, when you quantify the shoot dry weight, you see that in all cases, the rosette is bigger. Suggesting that somehow this optimization in the suberization in the endodermis is beneficial to cope with these nutritional stresses. And when we quantify the ionome, the concentration across this different combination of nutritional stress, we also identify difference, and you can see in this black line. So for example, in minus iron, for copper, you have a totally different pattern. In axenic conditions, you have low copper, but when you put the synthetic community, the concentration increase. So somehow the microbiome present in this system optimize the ionome through the optimization of the endodermal suberization. And this difference was quite dramatic in the case of sodium chloride and in the case of minus iron, as you can see in this PC principal component analysis. But there are another thing that we ask, this regulation affect all the mineral nutrients in the same way, or there are a particular sector of the ionome that is more susceptible to the microbiome control? So again, we proved that this effect that we were seeing using the synthetic community have nothing to do with fertilization, because the same experiment with the heat kill bacteria grouped again with the no bacteria. So they are not like a real perturbation when you add the bacteria, when you add dead bacteria. By the contrary, when the bacteria is alive, you see that the perturbation in the ionome, in the mineral nutrient accumulation, is dramatically different from no bacteria control. Surprisingly, when we analyze the concentration of these 20 different elements in Columbia plant with the synthetic community present, we identify a sector of nutrient that stop the oscillation when we remove the suberin in different lines. So we express the CDF, that is a suberin degraded enzyme, and when we don't have suberin, and we essentially see that this line with no suberin behave like essentially like Columbia with the synthetic community. And when we don't have suberin in this line and we add the synthetic community again, we see that these ions remain pretty much the same. By contrast, you see the perturbation here when you add the synthetic community, suggesting that this cluster of ions are more susceptible through B regulator through the microbiome pathway that control the suberization in the endodermis. Until now, we essentially demonstrate that the microbiome inhabiting the root have the capacity to modify the suberin, but also the Casparian street. But we don't have any idea, we don't have any clue how this microbiome pathway, this novel microbiome pathway, is connected to the existing signaling that we know that regulates suberization in the endodermis. To kind of understand the mechanism behind this interaction, what we did was a transcriptional analysis using RNA-seq, and we essentially select a couple of genotypes, wild type and MYD36, that is the main transcriptional factor regulated the deposition of the Casparian street and somehow related with suberization. And we did the hierarchical clustering analysis, and we identified cluster of genes, as you can see here, that essentially, whose expression differentially oscillate with the presence of one genotype, but also with the presence of the synthetic community, as you can see here. So all of these genes are induced only when the synthetic community is present, or repressed when the synthetic community is present. When we analyze the gene ontology of these genes, we essentially identify that all of these genes that getting repressed with the synthetic community or when the microbiota is present are essentially enriched in ABA. And it was quite surprised. It was like, wow. So it seems like these phytohormones is related with this signaling. So to prove that, what we did was identify a couple of mutants, one related with ABA signaling and the other one related with the ABA synthesis. And we analyze if the microbiome effect on suberin deposition is conserved in these mutants. As you can see in wild-type plants, every time you have the microbiota present, there are an alteration in the synthesis of suberin, but this alteration or this oscillation in suberin mostly disappear when you make a perturbation in ABA. By the contrary, in ethylene mutant, you see that this capacity of the microbiome to induce an alteration of a perturbation in suberin is conserved. So yes, that actually ABA is necessary and it's part of this mechanism. We also use lines, PCAS ABI, that we essentially interrupt the ABA signaling on the level of the endodermis. And again, after 5 days and also after 14 days, we see that the distribution of suberin in the root, in the case of the line as compared with the Columbia, is impaired. And we also see this after 14 days. To stress this idea that the ABA is involved in this signaling, we use a reporter line fused to GFP for ABA, and again, as you can see here, in different zones of the root. Every time you have the microbiota present, this signaling is getting reduced as compared to actinic condition, as you can see here. And here you have the quantification, it was significantly different. Suggesting that the ABA is important. So the optimization of this suberization in the endodermis is mediated by the repression of ABA by the microbiota. As a final model, we essentially demonstrate that the microbiome have the capacity to modify Casparian straight also suberization through the repression of the phytohormone ABA. As a conclusion, I want to stress that that's the first evidence that the genes regulating the root diffusion barrier influence the composition of the plant microbiota. We reciprocally demonstrate that individual members of the plant microbiome alter or influence the root diffusion barrier function, and it has consequences for the adaptation of the plant to environmental perturbation. And it's interesting that this effect is mediated by repression of the plant's ABA translational response. More importantly, we generalize the role of the microbiome in controlling root diffusion barrier across kingdom. And I don't know if you realize how critical is that, but we envision a lot of application of this, essentially in the development of new strategies based on the base of microbiome, essentially to induce changes or to modulate the suberin production in crops. And we anticipate that in the future, hopefully, we don't need to wait too much for this. We're going to see this strategy to try to modulate, for example, the capacity of crops for carbon sequestration, to increase the content of beneficial mineral nutrient or reduce the presence of toxic element. And in other words, to develop a more resilient and more environmentally friendly agriculture. Before I finish, because I think that I talked too much, I want to thank you to the Nottingham team that was directly involved in this project, but also our collaborator from the University of North Carolina, Bond University, Professor Julia Borjor and Paul for the bacteria collection. And obviously, thank you to all of you for your attention. This is our funding, the University of Nottingham, the Royal Society, and the Future Food Beacon of Excellence. Thank you. I can take questions.
Thank you very much, Gabriel, for your wonderful presentation and spectacular work. I really enjoy listening to you talking about what you guys have been doing. We already have some questions from the audience. So Ita is copying the questions and posting them in our chat here. So if you want, I can read them to you, or you can whatever you prefer. It's on you. So the first question is from Felipe Marasquim, a colleague. He asked, what would be the plant signals triggering the microbiome association?
In general, or in this particular case?
We should have a delay, but I think it would be like more in general. Maybe Felipe can add to that later, but I think it would be more. Yeah.
So this is a kind of a complex question, and it's quite context dependent. So I can talk in general. So we see that there are certain kind of compounds that promote the association. By the contrary, there are certain kind of compounds present in the root exudate that are detrimental for certain group of bacteria. So it depends on the context. But more and more we are learning about how this signaling happens. So colonization of the root or the shoot or whatever organ you are studying is one of the more, I would say, critical questions. I think that it's important to learn how to extract this principle to start using inoculum in agriculture, for example. So I'm sorry to don't have a more specific question, but this is kind of hot spot in the field. So that's why microbiome is so difficult, because we integrate a massive amount of other disciplines, including ecology.
Yeah.
This is something that is ongoing. I think that we need to figure it out how to get more ecology people involved in the microbiome studies in order to start extracting principle. To be honest, I would love to say, okay, it's happened like this and like that, and essentially it's these two pathways, but unfortunately it's not like this at the moment. It's more like a case by case.
Right. So Felipe Marasquin, he complimented that he's also interested in this case specifically. If you know some signals that could trigger that association. So if you can compliment on that.
There are a lot of papers. There are one in particular kind of recently published by the group of Paul Max Planck, describing this signal in the context of iron starvation, and it's very nice. It's basically mediated by scopoletins.
Yeah.
I think that last week or 2 weeks ago, there was another paper describing the role of flavones during nitrogen starvation and how these compounds modulate the root-to-shoot axis coordination. There are some signaling quite described, but I think that is case by case.
Yeah, we're still in the beginning, right? It's just too little. Okay, so there's another question here from Igor Cesarino from USP. He asked if it is possible that the differences in the root microbiome among the different mutants might also be related with the accumulation or reduction of metabolites upon blocking a metabolic pathway.
That's an interesting question. In fact, it was one of the questions raised by the editor. So we have in the paper a lot of control, massive amount of control. I decided not to include it here because it's quite distracted. So we quantify the level of hormones in the root. We discard that was not a question of ethylene and ABA concentration in the root. We also quantify a lot of developmental parameters, and we did a metabolic analysis. In general terms, the metabolism, at least the primary one, is not so dramatically different across the collection we used. But it doesn't mean that for certain compounds can be different. So this is something that I want to explore. So at the moment I'm working in try to define the regulatory elements involved in this crosstalk, and definitely metabolism is one of the aspects that I want to touch. Related with this particular question, I don't have any idea about it. It's an open hypothesis.
Okay, another question. Jonathan is congratulating you for the great work you did. And he asks if, in your view, how do these findings affect the study of plant nutrition and endodermis function, which is mainly carried out in axenic conditions? That's a key question, right? I mean, we're scared about that.
How this affect that?
Yeah.
I think that I expected to see like a massive amount of paper related with that, with this content. I think that at least I'm organizing a meeting at the moment, and at least 2 abstracts was relating with this topic. It's kind of confidential, but I anticipate that we're going to see more of this. Another thing that is not confidential is that the group of Joan Corey in UC Davis have a massive amount of budget, more than 40 million, I think, and they are studying how this crosstalk happens, so how they can modify sugarization in order to attract more CO2 from the atmosphere. And I think that at some point they need to address the contribution of the microbiome. I think that the microbiome is affecting not only nutrition, it's affecting defense and everything, development. And more we are learning about it. This is something that we cannot ignore. As you can see from this, that this is a small example of the things that we can do with the microbiome. And the arsenic response is dramatically different from the response when the microbiome is present. So it's like another world. So it doesn't mean that all the research conducted on the arsenic condition is not valid. By the contrary, I think that the majority of the principles defined under this condition help us to kind of add this layer of complexity. A good example is what I showed here. So the plasticity of endodermal suberization under arsenic condition is there, and it's important for the absorption of environmental perturbation. But I will say that it's a little bit incomplete, because on the nature and on the natural condition, this happens slightly different. So it's not as big as under arsenic condition. There are certain level of buffering effect, and the accumulation of a mineral nutrient is also distinct.
Sure, thanks. There's another question from Guido, a colleague of ours. He asked if we should stop washing and peeling our roots for consumption, because in other words, it would. He's asking if the root microbiome could be a source of our own microbiome.
Yeah, I think that not only the root microbiome, also the soil microbiome is. I think that there are at least recently a couple of papers describing this, how the soil microbiome affect or somehow is present in the gut. Yeah, there are a lot of, I would say, interaction between the gut and the soil microbiome.
It might be even it should be to a less extent, but not only from roots that we eat, right? Leaves and even grains. We should get some. Yeah, sure. Nice. Okay, there's another question from Rogério Marges. He's asking if there's any info about the effect of other hormones than ABA, especially auxin and brasinosteroids, that could affect root morphology and growth.
Yeah, in our previous publication in Nature, we described the role of ethylene and auxin, essentially more auxin in root development. This is already published. In the context of microbiome, for individual microbes, there are more publications, but definitely, at least for ethylene and auxin, we know how the system works. So we know that there are a genre called Barioborax that is essentially for the root development in nature and in the context of microbiome. And the mechanism is driven by auxin. And this is something that I would like to explore in the future. In fact, we have another project that I hope Felipe gave me the opportunity in the future to talk about it, and it's related with lateral root formation. So we're going to be addressing the role of auxin in lateral root formation.
But not brasinosteroids, right? That's not included in the
steroids. I know that there are people working on this, but I don't remember now something already published. But I know that there are at least a couple of groups working on it.
Okay. Jeanette Fetus asking if do you see some kind of pattern that could allow to predict which nutrients will have higher or lower shoot concentrations more suberin in the Casparian strip? Well, it's actually
for individual isolates, I think that we can predict somehow because the correlation is strong. In the context of a natural microbiome, is more difficult. Essentially because the ionome is a very complicated network. I think that Felipe knows more than me about it, with a lot of buffering, interaction, a lot of interconnection. There are a lot of chemical similarity. I think that before make a prediction, we need to make a big effort in order to define the ionome network and then try to understand how this network functions essentially in absorbing environmental perturbation like the microbiome. I particularly found the ionome field very, very difficult, and sometimes we cannot predict this interaction easily. Hopefully in the future we'll be in a better position. Individually, I will say yes. In a more complicated context, I will say it's a challenge.
Okay.
Figure it out.
Yeah. This was the last question from the audience, but I have one question of my own. If you can answer that. You mentioned in the beginning of the story that you told about that you're measuring also the microbiome. You're checking the microbiome in the shoots, right? But of course, the submarine story is all concentrated in the roots. But do you see any way that the shoot ionome could be controlling the nutrition of the shoot itself? Do you have anything going in that direction? Because I mean, I know that the microbiome might be less complex. I'm just guessing in the shoot, but it feels that if they're in the same place, they could also be interacting, and one could be controlling the other. So how do you see that interaction?
The correlation we observe between the ionome and the root microbiome is also present in the shoot. It's less obvious, but it's significant. So my expectation is that the shoot microbiome can contribute also in a different way, but also at the moment I'm exploring this line in maize, and we can see so far is like, don't put this on Twitter, please. Individual leaf have an individual microbiota, also an individual ionome, and there are certain level of interaction in there that determined nutrient recycling and also nutrient accumulation. So they should have also something to say. I think that this is a kind of trendy topic at the moment, the root-shoot axis in plants. So they are light, they are metabolic communication, and I think that they are a very nice review from, I don't remember exactly the group, about the root-to-shoot axis that kind of address this question, Philip.
Okay, I'll check that. Okay, if we don't have any other questions, I would like to thank you once again for accepting the invitation to be here. It's
hopefully in the future we can do this activity again.
Yeah, sure. Yeah, it'll be a pleasure. We should meet in person and
in both directions.
Sure, definitely. Thank you very much. Give my best to everyone there, and we appreciated your time to talk with us and present your work. Thank you very much, and congratulations for the great work you've been doing.